Hello, today we are going to dive into cell division, specifically mitosis, and we're going to touch on cytokinesis. We talked about this a little bit in our last set of notes, but now we're going to really dive into it and really get into the nitty-gritty of how cells divide. Before we actually get into cell division though, we need to talk about the different forms of DNA that will show up in the different stages of mitosis. Okay, so there are two forms that we're going to focus on. The first one is chromatin. Chromatin is long and thin, right? Tin rhymes with thin, so that's a good way to remember that. But basically, chromatin is just unwound pieces of DNA, like strands of DNA, that exist in cells. You can think of this as like a strand of spaghetti, right? You have a bunch of spaghetti in a bowl, all of that is long and thin. It's going to remind you of chromatin. The second form that we're going to talk about is chromosomes. And chromosomes are maybe what you're more used to when you think about DNA, but chromosomes are condensed or coiled. So going back to our idea about like a bowl of spaghetti is filled with chromatin. When you stick a fork in it and you twist that fork to get all of that spaghetti on your fork, when you're coiling it around that fork, that's how chromosomes are formed. So chromosomes are condensed, they're tightly wrapped, and you can see them under a microscope. Whereas chromatin is sort of long, thin, unwound, and you can't see it under a microscope as easily. Okay, now that we have our two basic forms of DNA, let's get into actual cell division. So the general purpose here is to grow new body cells or repair and replace old body cells. Okay, so when your body cells get old and tired and they don't work as well, that's what cell division is there for. It replaces them and makes new body cells. Now this image should look familiar to you, but interphase is the first part of the cell cycle. Just as a quick review, it's the first stage, and DNA is in the form of chromatin, right? That long and thin piece of spaghetti. In this stage, DNA replicates and the cell grows. This is where the cell spends most of its time, 70 to 90% of its time, and this is sort of just like the precursor to cell division. Now, mitosis is the second stage of the cell cycle, and this is where the cell actually divides. Well, the nucleus actually divides, I should say. So we start with a parent cell, and it produces two genetically identical cells. There are 4 phases throughout this, and we're going to talk about each one, but just as a little like preview of where we're heading, right? Our first one is prophase, then metaphase, then anaphase, and then telophase. In prophase, you can remember this as the preparatory stage, right? Because it starts with a P. In this stage, the nuclear membrane dissolves, right? So our nucleus is starting to dissolve, and chromatin, right? Because in interphase, the DNA is in chromatin, but in prophase, that chromatin coils into chromosomes. So now we're going from chromatin into chromosomes, and centrioles, which are these little organelles up here, they shoot out spindle fibers, right? They're kind of like Spider-Man here. They shoot out like these web fibers, which will be used throughout the rest of mitosis, okay? So big piece here, nuclear membrane dissolves, chromosomes start to coil, and spindle fibers start to form. In metaphase, chromosomes meet in the middle, right? And metaphase, right? Hopefully it helps. In this, it's pretty straightforward. Those spindle fibers that came out of those centrioles in prophase, they attach to the middle of a chromosome, and they move those chromosomes to line up down the middle of the cell. And that's the whole stage. That's all that happens here is those chromosomes now move to the middle of a cell. In our next phase, we have anaphase, where chromosomes move apart or away, whichever one is easier for you to remember. Now, our spindle fibers that were shot out are still connected to the chromosomes, but now they're starting to retract back into the centrioles. So what's happening here is that those chromosomes are being ripped apart from each other, and they're moving into opposite sides of the cell, right? So they're being pulled away from each other. This is so that we have one copy of each chromosome in each side of the cell, right? Because we want two genetically identical cells at the end. This is how we get that. In our last phase, we have telophase, and two new identical nuclei are produced. Telophase is sort of like the opposite of prophase. So in this one, our nuclear membranes start to reappear. We now have two brand new nuclei that are identical to each other, and so the membranes reform to make an actual nucleus, and the chromosomes begin to uncoil back into chromatin. So, no longer considered a chromosome, begin to uncoil into chromatin. And then our last stage, this is the third stage of the cell cycle. This is not technically part of mitosis, this is part of the cell cycle, but it's not mitosis. Mitosis is just prophase, metaphase, anaphase, and telophase. In cytokinesis, the cytoplasm actually divides. So this is where we go from telophase on the top into two actually separated cells. And they're identical, but the cytoplasm has finally split, so we have two distinct cells. Now, cytokinesis is different in animal cells and plant cells because there are different organelles in animal and plant cells. So in animal cells, because there's just a cell membrane around the cell, what's called a cleavage furrow forms, and basically it's the cell membrane, and it starts to pinch in and keep pinching in across these two cells, and eventually it will pinch itself off, and so then it will form your two new cells, but it's because the cell membrane is so flexible that it's able to do that. In plant cells, they have a cell wall, right? And that cell wall is super structurally tough and strong. And so instead of being able to just sort of like pinch in and split the cells, a cell plate forms, and that is basically like a mini cell wall that forms in between the two cells, and eventually it will divide the cytoplasm completely, and you will end up with two brand new identical plant cells. All right, that's it for today. Great job. I'll see you in class.